All right, so before we start Unit 6, which is all about genetics and inheritance, we're going to talk about DNA. We've already talked about DNA, but we're going to talk about it more in depth in terms of like how it applies to how you get it from your parents and what exactly happens to make you look and function the way that you look and function, okay? That's what Unit 6 is all about. Inheriting genes from your parents, how they get expressed, why you are the way that you are, okay? But before we get into that, because it's been a little bit since we've talked about this stuff, you need to know this stuff here, which is about DNA, proteins, traits, and genes. Some of the stuff we haven't talked about, for a while, and so I just want to recap that before we jump into Unit 6, all right? Because this is all going to come back into play. Just because you learned something in the past doesn't mean that you don't need to know it anymore. It all builds on itself. So the things I want you to know before you go, if you don't already, are what are genes? How do genes control protein production? What molecules control your traits? And what is an accidental change to the DNA or the genes called? What is that called, and how does it affect those traits? Okay, so let's start off by talking about DNA again. Now remember, if DNA is made up of these things called nucleotides. And remember that a nucleotide is made up of a phosphate, a sugar, and a base. Now, these bases, there are 4 different kinds in DNA. We call them G, C, A, and T. So on the other side, remember DNA is double-stranded. Okay, so across from every one of these bases, you have other bases connected by hydrogen bonds. Oh, sorry. Okay, so this is one strand of DNA. It's got a phosphate sugar in one of these 4 bases. It'll be like phosphate sugar G, phosphate sugar C, phosphate sugar A, or phosphate sugar T. And then across from that on the other side is the same thing. Only it'll be like this. It'll be in reverse order, right? It'll be should have gave myself more room, but whatever. On the other side, it'll be phosphate sugar C if there's a G across from it, or phosphate sugar G if there's a C across from it, phosphate sugar T if there's an A across from it, and phosphate sugar A if there's a T across from it. So A and T always go together, right? And G and C are always across from each other. Okay, these are called the base pairing rules. G always with C, A always with T, okay? So if you have a segment of DNA that is G, C, C, T, T, A, if that's strand 1, well, across from it on strand 2 will be, what will be here? Across from G is C, G, G, A, T, right? That's DNA in a nutshell, okay? So remember, that's how DNA works, and the instructions for how to make everything in your body is encoded in this DNA. So there's actually code, just like computer code, that if you look at it doesn't make sense if you don't know how to read it. But once you know how to read computer code, or someone who's really good with computers can read the code and actually know what it says. And there is a way to decode this and know what it says, which we're about to get to. Okay, so what this does, though, is, like I said, it does provide the instructions for how to make and do everything in your body. But the molecules that make and do everything in your body are proteins, right? So DNA is where all the information is stored on how to make the molecules that control your traits, which are proteins. But it does not work this way. It doesn't go straight from DNA to proteins. If you'll remember, DNA, the information in DNA is first copied into a strand of what we call mRNA. So that M stands for messenger. And that's all it is. It's just the messenger. The mRNA, the messenger RNA, just copies the instructions from the DNA and delivers them to this place called the ribosomes, where the proteins are made. Okay? So before your traits are expressed, proteins control your traits. Proteins control your traits, but mRNA is what delivers the instructions from the DNA to the proteins. Okay, so this first step is called transcription. Remember, it's a two-step process. Step 1 is called transcription, and step 2 is called translation. I know the board is getting messy. I will erase this soon, okay? So transcription and translation are the two steps. When you go from DNA to mRNA, when that information DNA is copied into this, that's transcription. And when the mRNA is used to make the protein, that's called translation. The function of mRNA, I already said that. That's what that next question is, but you've got that. mRNA, remember, just carries the message, the instructions from DNA to the ribosome where the proteins are made. So whenever this happens, though, let's go back to the protein synthesis, the DNA to RNA to mRNA to the proteins. Okay. Again, transcription, translation. Okay. What happens is that you have this strand of DNA, like G, C, C, T, A, T. We're just going to make something up. And what happens is that this is your DNA strand, and this gets copied into mRNA, right? So that G gets copied into a C. The C gets copied into a G. We're not making an exact copy of it. It's not like we're copying and pasting, remember. We write the complements. We write a C instead of the G. We write whatever goes with that original DNA base. We don't just copy the DNA base. So T gets copied as A gets copied as T gets copied as A, right? So this is your new mRNA strand here. And then the mRNA strand gets used to make the protein. But how do we do that? Well, remember, you got to break it up into groups of 3. And each group of 3 is what we call a codon. So we have codon 1 and codon 2, okay? And each codon codes for the production of an amino acid. So this will make an amino acid, and this will make an amino acid. But there's another guy you need to remember. His name is tRNA. tRNA is the one that actually brings the amino acids to the ribosome to put them together to make the protein, right? So tRNA kind of looks like this weird little guy like this. Okay, and this thing up here is the amino acid. Remember I told you that tRNA is kind of like a plug. When you plug it into the socket, it has those 3 prongs on it, the top 2 and then the bottom one in the middle. Well, the tRNA, which is this guy here, he kind of plugs into each one of these codons, depending on which codon it is, and he'll bring that amino acid for that codon and plug into these 3. Notice how there's 1, 2, 3, 1, 2, 3 things here, and there's 1, 2, 3 bases on mRNA. Well, these 3 bases on tRNA will plug into these bases here. Oops, I just realized that I put a T instead of a U. Remember, there is no T in mRNA. In RNA, there's a U instead. I was going too fast, I'm sorry. But that little guy is tRNA, that's who you need to know. And he's helping to build the protein because he's the one that actually delivers the amino acids you need to make the protein. The way that you find out what amino acids are made by this strand of RNA, or you use a codon chart. We talked about that. I'll let you guys remember how to use a codon chart. I don't have one to show you right now. But the question provides you with a codon chart. You can use that, okay? You just look up the 3 bases. All right, so that's how DNA turns into proteins, and proteins control your trades, right? But here's the question. Then we talked about how even though all cells so let's say this is a brain cell, this is a heart cell, and this is a lung cell down here. All these cells have the same DNA, okay? They all have the exact same DNA. So why is this one a brain cell? Why is this one a heart cell? And why is this one a lung cell? Well, it's because even though all these cells have all the same DNA, in this cell, only the brain cell DNA gets turned on, or what we call expressed. This is what we call gene expression. Remember, a gene is just a short segment of DNA. So if this is your DNA, remember it looks like a ladder if you untwist it, or if you want to look at it twisted, you can look at it like this. So a gene is just a short segment of DNA. So that could be gene 1, and maybe this is gene 2, and then maybe this is gene 3. So maybe gene 1 is the brain gene, right? So in the brain cell, only this gene gets turned on and the other 2 genes are left off, kind of like light switches, okay? That's called gene expression. So different genes get expressed even though all the 3 of these cells have this full set of DNA with all, I said DNA with the 3 genes. The brain cell and the heart cell and the lung cell gene. Only the brain cell genes get expressed in the brain cell or turned on. Only the heart cell genes get expressed or turned on in the heart cell, and only the lung cell genes get expressed or turned on in the lung cell, okay? So they have all the genes, but not all the genes get expressed. So genes can be expressed at birth and you can't change them. But then there's some genes that get expressed, can be expressed, meaning they can be turned on or turned off later on in life as you go throughout life, depending on the situation. Okay? So one of the examples that we talked about was with the Himalayan rabbit and how in the winter its fur turns black and in the summer its fur turns white again. Well, that's because there's black hair genes and there's white hair genes. And in the winter, the black hair genes get turned on and the fur turns black, but then when it gets hot again, the black fur genes get turned off and the white hair genes get turned on. Okay? So temperature, things like temperature, or that's not it, there's other things too. Things in your environment can make your genes turn on or off as you go through life, depending on the situation that you're in. It's part of the adaptability and survivability of your body to be able to do those things and change depending on the environment to increase your odds of survival. Okay? It's just part of what we've learned how to do over millions of years of evolution. And some traits are what we call sex-linked. Okay? So remember, your cells, your DNA is broken up into chromosomes. So DNA is not one solid piece. You can it's actually many small chunks that we call chromosomes. Okay? So let's say that each one of these is a chromosome. It's just a shorter segment of DNA. Your DNA is not all connected together end to end in one solid piece. So let's say this is a chromosome, this is a chromosome, and this is a chromosome. Okay? Now, on these chromosomes are the genes. So each of these chromosomes, let's call them chromosome 1, 2, and 3. You can break these up into even smaller sections, like that one, and that one, and this one. Maybe this chromosome has 3 genes, gene 1, gene 2, and gene 3 on that chromosome. So some of these chromosomes are what we call sex chromosomes. Your body has your cells, each cell in your body has 46 chromosomes. And 2 of these 46 are what we call sex chromosomes. So let's say that these 2 here are sex chromosomes, okay? When I say sex chromosomes, I'm talking about male and female. Okay? So male or female. And in males, your 2 sex chromosomes are what we call XY, and in females, they are XX, okay? So like I said, you have 2 sex chromosomes. If one is X and one is what we call Y, you're a boy, and if they're both X, you are a female. Now there are some traits whose genes, remember because genes control traits, there are some traits whose genes are located exclusively on one sex chromosome or the other. Like the calico fur on a cat. It's located on the X chromosome, which is why female cats are much more likely to have that trait of being calico fur. Because it's on this chromosome, and females have 2 X chromosomes, whereas males only have 1. Okay? So that trait could be somewhere in these chromosomes. If they're both X, then you have double the possibility of having calico fur. Okay. So when you change the DNA, because DNA gets copied into mRNA, which gets copied into amino acids, which when you put them together make proteins. I'm just repeating what I said earlier, but in a different way. And proteins control your traits, right? So if I change the DNA, I change the mRNA, which changes the amino acids that get made, which change the proteins that get made, which change the traits that you end up with, okay? But it all starts here. That's where the chain starts, okay? And when you change the DNA, this is what's called a mutation. Now, mutations can be bad or good, just depending. Some mutations are bad, some mutations are good, okay? But there's different kinds of mutations. So let's talk about what kind of possibility that changing the DNA could have, okay? So let's say, once again, that this is your DNA strand, okay? We're just looking at one of the two. We don't need to look at both strands, all right? So if this is what it's supposed to be, let's call this the original, or OG for short, okay? The original strand of DNA. And I make a copy of this. I want it to be an exact copy, but let's say that instead of making an exact copy, I have a typo. So, oops, I put an A when I should have put a C, okay? Well, that A shouldn't be there, but this is what we call a substitution mutation. So when I change the DNA, I'm changing the RNA, which changes the amino acid, which changes the trait, right? Well, a substitution isn't that bad because it just changes this piece of DNA only. The rest of the DNA is still good. It's still TAT like it should be after that, right? And this G is still a G. This TAT is still TAT. It's fine. Like, it's not that big of a deal. It's just, we're just changing one amino acid. I mean, it is a big deal, but it's not as bad as the one I'm about to show you. But let's say that instead of doing that, I accidentally, let's say this is that's the sub there, substitution. But let's say that instead of just making a substitution, I actually accidentally inserted the A. I plugged this A, I took an A and I plugged it in here. Well, when you do that, it moves every single letter down one. So now, not only is this codon thrown off, instead of being GCG like it's supposed to be, it's now thrown off. But this set of three, or this codon, is also thrown off. So you see in the substitution, the second codon, TAT, is still like the first one. It's still the same. Well, in this one, not only is the first codon thrown off, but now the second one is thrown off. The one after that will be thrown off. And this is what we call an insertion. So the insertion mutation, or a deletion mutation, which is just the opposite, if I had deleted one and everything went back one, throws everything after that point off too. So this doesn't just change that one codon, it changes every single codon in the entire sequence after that. So this one is much more, insertion and deletion are much worse of mutations than a substitution. Okay, you have a question about that in there as well. Okay. And the last thing is that these mutations are typically not passed on to offspring. You have what's called, okay, let me just keep it simple. For a mutation to be passed to your offspring, it has to happen in what we call gametic cells, which are sperm and egg, okay? These are the gametic cells. Every other cell in your body that's not a sperm or an egg cell is what we call somatic cells. This is every other cell. Okay? So if you have a mutation that is passed on to your offspring, it's because it happened somewhere in either your sperm or your egg cell. If it did not get passed on to your offspring, then it happened in what we call one of your somatic cells, which is just another cell that's not your sperm or egg cells, okay? All right, so in summary, DNA controls your traits, but it doesn't go directly. DNA gets copied to mRNA, which makes amino acids, which makes the protein, which makes the trait, okay? When you change the DNA, you change the traits. Even though all cells have the same DNA, they don't all get expressed. That's why some cells turn into one thing and some cells turn into another thing, and that's called gene expression, okay? And when you change the DNA, it eventually changes the trait. That's called a mutation. And that is everything that I wanted you to get caught up on before we move on. Okay. Thank you guys. Y'all have a great day.